if I'd known how much that everybody had liked disaster photos, I could have included the last one I had before I got on a plane, which was where my 4-year-old discovered the wonders that happen when you mix toilet paper with water, and he redecorated our bathroom in a 20-minute space before I left. So if I can incorporate that into illustrating soil health benefits, I will try and do that. So look, to kick off there again, it's great to get an opportunity to speak again today, and just to, can you see that? There you can. So just to start and tell you about our journey in terms of our soil health. So it started a number of years back because both myself and my husband were vets, as I said, and we tend to have a very science-based approach to how we run the dairy farm. And what we noticed was that over the years, our farm grass production, when benchmarked against other research farms, it didn't seem to perform as well as it should. And our pH and soil indices were all in good shape, but at the same time, we still weren't growing huge tons of grass. And believe it or not, it took quite a while for us to realise that our soil was probably at the base of it. And then we said to ourselves, well, what do we do about learning more about our soil? So my husband took on a master's in environmental sustainability and ended up doing a thesis on our farms, and I went to the local university and did an entire course on soil health and water management. So yes, you get the picture now. Both of us are complete nerds. We're science geeks, but we love it. And what we learned, I've condensed into simple steps for you guys to learn from. So firstly, point one, and this is how that works there. Is that after kicking in? No. That's our beautiful farm, but we need to move it on one tick. Yes. Know your baseline. So they're my kids lying in the grass there, but know your baseline. And we've covered this a lot over the last couple of days. This is what's your starting point, because what you're working with in terms of your soil is so important. Soil is a living entity, as we've all learned, and then as it is a living entity, that means we have to deal in the physics, the chemistry, and the biology, okay? Because that's what composes our living entities. So I was always taught to focus on the chemistry, and that there were really, as a farmer, there were no other important measures. It was just soil chemistry. And if we focus solely on soil chemistry, we might end up, and we ignore structure, we could end up expecting a really, significant amount from soils, but those soils might be as compacted as rock. So, so the right. We need to get a more holistic view and a more holistic approach to our soils, basically. But first and foremost, you need to know your baseline. So, step one, we move to the physics part of it. Oh, that's moving on. There we go. So, step one is soil physics, and we've covered a lot on that over the last couple of days. And I was going to really get into it, but it's important to know what your soils are made of, and how that. Even your place in the landscape can influence what your soil's ability can do. So for us, we're on a sandstone formation. Sandstone tends to confer, a free draining soil, and so that's at the very, very bottom. So knowing your soil type. Is your soil type a productive brown earth? Is it a podzol? And all of this information is available to you on your soil information system. And you can, so and I checked. It's available in Victoria, I've checked. So this information is all out there for you to start up on and to click on your area, find out what type of soil you have. And now you're going to know, we spoke about soil texture as well, and that's all available there in your soil information system. So again, just, I can't emphasize how important it is that information's out there. And when we move on, that's step one. Here we go. When we move on, again, this is the type of soils that are available on our farm, and you can see on the left we have some brown earth soils that have nice structure. But again, looking at the other soil, which is a lithosol, a stony soil, we can't expect the same results from both of these soils, and you have to realise that about the farm, the soils on your own farm. Again, when we move into soil physics, we want to look at soil texture and structure, and what are the field site tests that we can do as farmers to determine this. So one is soil structure. We spoke about the slate test, so here we go. This is a slate test on the left. So what I'm after doing on our farm, slate test every single paddock on the farm. Very, very simply, that's a glass vase that I had. I got a net from onions, dried out a clod of topsoil, you dry it out, and then you immerse it into the water. And as Pauline was telling us the last couple of days, so basically what you want is that fundamentally, the foundation of good soil centers around soil structure. So, and the important thing is that that soil has to remain aggregated in the face of erosive forces. So that's the really, really important part. And what we're doing here is we dry out the soil, then we immerse it. So it's like, I guess it's like, a dry period and then we get heavy rain. And you want your soil to stick together, just like the toilet paper on our ceiling. So you brought it in there. You want it to stay together, but you don't want that soil to start to crumble apart, because your soil needs to be agglutinating together. That's really, really important. And so that's a simple test you can do across your farm. Again, the ribbon test, as we spoke about quite a bit, to look at your soil texture, do your ribbon test, and figure out how much the soil silt or sand silt or clay is in it. The next one, and I won't get into detail on that because I think Pauline has covered it a fair bit of late, but the next test is called the grass VES. And this has been, I think it's developed in Ireland, but VES stands for Visual Evaluation of Soil Structure. It's very, very, very simple. So you follow this infographic, you dig up a clod of your own soil, and it's a simple scoring system to look at soil structure by scoring the top of the soil, which is your upper root mat, and the lower soil portion, and you score it for structural quality. And it's such a simple thing to do. Again, I did grass VES on the entire farm. Grass VES, I did the slate test, and it gives a baseline for soil structure across the entire farm. And you can see then, is there areas for improvement or not? And it also shows up how management can impact on your soil structure. So if there's a lot of poaching going on, you might get surface compaction and things like that. So these simple farms, field site tests can be done by you guys and just, extend it across your whole farm. So step 2 is to look at soil chemistry. And we're all fairly well versed in soil chemistry. Your pH, your P and your K. And the big one is obviously pH, because what we call it at home, pH is your master variable. At home, we call it the cheapest fertilizer you can use. Because when you get your pH in the right range, it has a huge impact on nutrient availability, especially nitrogen. And that target pH for establishing swards, so for ryegrass about 6.3, but for clover, it's a bit higher at 6.5. So that's really, really important. I know we spoke about what's the appropriate range, but you need to, if you want to get legumes, you need it 6.5 and above. So just, and lots of research at home to back that up. Again, in terms of micronutrients, and so again, this is what we all have this is again our farm and what we've looked at. You can see pH in that upper, above 6.5 mostly across the farm, and our P's and K's. So, everyone's used to looking at that, but what we did is we extended out the testing a little bit to look at micronutrients. And that's an area that not hugely familiar with, but we're moving in that space, So what was a surprising finding there was that our soils are quite deficient in micronutrients. And if you look over at the far right, you can see cobalt, and we're pretty low in cobalt. But for example, cobalt is very important in rhizobium bacteria. It's a cofactor for that bacteria to work. And that bacteria is a nitrogen-fixing bacteria that works symbiotically with legumes, with the nitrogens in legumes. So in terms of nitrogen fixation, hugely important. And when you need to look at targeting micronutrients, these are areas that you can move into. Again, looking at that chemical testing and extending it out towards your organic carbons and percentage organic matter. This is a test you might do maybe once every 5 years or once every 10 years, just to see are you moving in the right direction. Because in terms of organic matter, that is the sponge of the soil. It influences water holding capacity as well as soil structure. And for example, a soil of 4% organic matter can hold more than twice the water of similar soil with just 1% organic matter. So knowing this will confer, it confers that degree we say drought resilience or drought resistance to your soils, depending on where your organic matter is standing. So step 3 is of course biology. And soil is one of the most complex habitats on Earth. It ranges, we have the microscopic, we have the macroscopic, and we have the mesofauna that we heard about yesterday. As farmers, what are the field site tests that we can do? Well, we can measure earthworms. Earthworms are known as the intestines of the earth, and that denotes their importance as keystone species. So they're involved in nutrient availability, aeration, drainage, and soil formation. So an earthworm count is a great starting point to roughly assess to reflect your soil biological activity. So I use the same size spade every time I go out. It takes 5 minutes. I dig the whole farm again, and you just dig in, measure your numbers, and assess, have you got adults or have you got juveniles, or what size they are. The adults, of course, have that little saddle on their back. Juveniles don't, so it's easy to tell them apart. The best time to do soil count or worm counts is at the shoulders of the year when there's moisture in the soil. So I do this in March and September. And of course, needless to say, the highest worm counts that we found were under multi-species swards and the heavy clover swards. So now you have checked your soil structure components and your makeup with your soil pits or your slate tests and using your grass vests. You have your chemical soil results, and you have a few more extended chemical testing done on the farm, and then now you have your worm counts. So what's next? And what is next is step 4, which is, and these are worm counts from last time, but step 4 is to make a plan. And you can see there again in this photo, if you look at, this is the soil underneath the multi-species swards. Do you see all those little balls? Like an incredible amount and variety of them, which are obviously worm castings on our multi-species swards. Like it is really incredible. So what did we do when we made a plan? We started to put in our multi-species swards and we wanted to, first and foremost, as I said yesterday, we started multi-species on the most drought-prone parts of the farm to bring roots into the ground, especially in those shallow soils. And for fields that we identified with compaction issues, so like that field on the right, that field had low worm counts. We put in the diverse grazable summer crops with fodder radish and stubble turnip, as well as phacelia and all those other different mixes. So what was said earlier was interesting. People mentioned the deep ripping, let's say, for improving soil structure. But the thing about that, the why of that is very important, because when I was on my soil health course, we were told that steel cannot solve structural issues in your soil. And we tend to use steel to loosen up structural issues in our soils. And what was interesting was said in the last talk was, well, you loosen it up, but you're putting in roots or you're putting in an amendment. And the why of that is that a root or an earthworm will form a stable chamber down through the soil. And it's these stable, these stable holes or cracks or infiltration roots that provide that, that bring that aeration down into the soil. Whereas when you rip it and you apply the same management, it collapses and goes back to exactly how it was. So knowing the why roots or earthworms is really, really, really important. And this is what it looks like. So that used to be a quite compacted soil. Look at the big, that's a deep burrowing earthworm. Look at the roots. And always when you think about soil, think about chocolate, right? Why do you think about chocolate? Well, cocoa powder is not what you want. You don't want a block of chocolate, instead you've got the other way. You're looking for chocolate cake, right? Chocolate cake, a beautiful crumb texture, that beautiful rich dark colour. Something that has aeration through it and holding together. So anytime we have groups in the farm, chocolate cake, that's what you're thinking of when you want to look at soil, okay? And you can see it there, you nearly eat that. Well, one of my kids might, but so you can see the benefits there. Roots, earthworms, look at the colour of the soil, you can almost smell it through that picture. And that was simply put onto a pasture that had fairly compact structure in it. Again, the benefit of multi-species, these two fields are 10 feet away from each other. They just had a really intense rainfall event. Look at the difference. I mean, it's chalk and cheese. Infiltration, drainage, and structure all improved with multi-species swards. Same day, 10 feet apart. I mean, it speaks volumes. So we changed to min-till for reseeding. And again, this is to reduce the impact on soil structure. We have very, very light soils on a hill. The tractor's upright this time, so we're in the right zone. So why did we do that? Because there was incidences where your field is prepped for reseeding, you've harrowed it, you have a beautiful soil, a soil bed for seed, you put it in and you roll it, and now you get a really intense rainfall event, and the rain is running a rush across your field and taking hugely valuable soil with it. I mean, it's loss of a natural resource that we can't afford to lose. So you can see there, glyphosate to kill off the sward. It was grazed tight beforehand and then direct drilled in, and a super result after that. We put a much greater emphasis now on composting farmyard manure. So it's rich, it's crumbly, and we target that on fields that that have lower organic matter levels. So that's been a big change for us. These are my sons here. We planted almost 2,000 hedge plants in and around the farm to create line breaks for water flowing downhill, to slow water velocity, so it has less of an impact on our soils. We are experimenting with seed dressing using mycorrhizal fungi. You can see the little web-like fine network of roots associated with the plants there. Can you see them? They look like a little web. So mycorrhizal fungi produce a secondary web rooting network that can scavenge nutrients and water. And we find that it extends the root network for seeds that are trying to get off, and trying to kick off and trying to germinate. And if a dry spell hits after sowing, you've got a greater root network and greater availability to scavenge water nutrients when it's needed the most. So for the important aspects of this talk is to find your baseline, do some testing, identify your issues, and put some practical corrective actions in place. We can't solve soil health overnight. This is a long-term game. So the most important thing is that identify your issues, but put the right action at the right time in the right location. And you're going to incrementally drive soil health improvements over the long term. Thank you.
Oh, thanks. Hello, everybody. Wow, what a view. What a crowd. I'm going to start by changing the slide. And I want to make a point about interactions. We can't, I think in the field I spoke about the importance of soil structure. What I was really talking about was the importance of aggregate diversity and how that links to plant diversity, which then links to soil diversity. So it's pretty pointless trying to drag them apart and look at them separately when you're working in a system, and you really need to focus on the interactions between those things. As a soil biologist of many years, I've found that I have to have that additional information, which we call the halo of information around the soil biology, so that we can make sense of what's going on. So we've always had to integrate data sets the soil physical, chemical and some biological data sets together to really understand what's going on with soil biodiversity. So when we consider biodiversity is a component of soil health, but when we talk about biodiversity, we are really talking about a community or an assemblage of bacteria, fungi, archaea, mesofauna, or microfauna, mesofauna, macrofauna, all those things that Gillian went through and also Lynn went through yesterday. When we talk about the community, we're talking about it with respect to where it's located. So when we do that, we're talking about a microbiome. In dairy and grazing systems, where there are probably 3 microbiomes of importance, I think I've called them champion microbiomes, and trying to make a point there that they're very important to our system. So we all know about the rumen microbiome. There's also a plant rhizosphere microbiome, so around the root of a plant, of different plants, there are different populations or microbiomes, and you'll hear a little bit more about that later. And then you come down to the soil microbiome. The soil microbiome is probably, well, it is the most diverse ecosystem on the planet. So as Lynn pointed out yesterday, it's highly complex. There's lots of interactions, there's lots of food webs and feedback systems going on. But the point I want to make about this slide is that we have control. We can manage, harness, exploit those different microbiomes through the way we manage our systems and through the inputs we use. For example, in the rumen, there's plenty of things we can do to the animal's diet, the cow's diet, to change the mix of species in that location, and to prevent or to slow down methane producing organisms. And this morning I saw there was a new product on the market that is being, I think, researched by CSIRO and Sauvignon Orange, and they've just come up with a new product based on a soil fungus and the products, the peroxidases that come out of the soils, and slow down the methane producing bacteria in the rumen. So there's manipulations you can do there. Obviously with the plant biodiversity, you can change the types of plants you grow, and each plant will attract or recruit into its environment a specific group of organisms which help it to do things. So help it accumulate nutrients, protect it from, or protect the plant root from disease organisms, and also increase the absorptive capacity of the roots. And then you come down to the soil, and we've heard over the last couple of days all the different things you can do to that, all the inputs that go in there in our normal practices, and included in those are addition of fertilisers, alternatives to fertilisers, pesticides, grazing intensity, all those things we can put into the disturbance category. All those things will have an impact on the microbiomes. Stuck. Oh, there you go. I want to make probably two or three points about that cover off on some of the principles around soil biodiversity. The first one is soil biodiversity is made up of what's there, so the structure, the different groups, and that's depicted by the wheel here. The reason it's a wheel is because it's really a histogram rolled up. The histogram's too big. So that's what is there. So that's called structural biodiversity. In most of our discussions, what we think about is structural diversity. We think about who is there, what groups are present, and give them names. More important than that is functional diversity, the roles that these different groups play in the soil. And these roles or functions are most relevant to us in our dairy systems. For example, we've heard all about nitrogen fixation, phosphorus solubilisation, hormone production, suppression of disease. There's a whole range of roles. So I think someone asked me the other day, what measure would you take if you were given one to take in the soil? And I think off the top of my head, I said functional redundancy. And yeah, I really should have said penetrometer resistance. It's still bothering me. But so what functional redundancy is if you look around that wheel, you can see there's a repetition of the different functions. So different groups of organisms can do the same thing. So with if you look at nitrogen fixation, that can be carried out by a group of organisms. So that if one group is knocked out, it doesn't mean the whole functionality of that, the nitrogen fixation will be knocked out, because another group will pick it up and off it goes. So that's how we measure that. That's what functional redundancy is. And it makes our systems very resilient to disruption. So the whole thing doesn't collapse overnight because we get rid of one species. So that's the importance of biodiversity. Having lots of species that can do the same very important process in the soil. So I don't want to labour this point, but I will. We can't do much about soil texture, the proportion of sand, silt and clay. We've got to live with it. What we can do though is manage the soil structure. So those particles and how they form together depend on organic matter. And so we can manage how much organic matter goes into the system, the quality of the organic matter, the timing of the organic matter, where it goes, the distribution in the profile. We can actually manage the soil structure. So I think this is really cool because I don't know if it's in the same order, but I thought, well, how do we incorporate biodiversity into our thinking? And it is cool because it aligns with some of the things that Gillian just said before about the need to develop a baseline, the need to see who is there, what they're doing, and maybe record it and record when you make these observations. The second thing is to consider our options. What options do we have now? I've heard all sorts of options put on the table or presented over the last couple of days. So we need to consider those. And the third thing is we need to act. And whether it's just not shotgun, it's very incremental and we build and build and build, that's really good. But we've now got a decision support process, but I would challenge people to consider biodiversity, soil biodiversity, in that decision process. So I wanted to get to the crux of what I need to communicate to you, and that is to consider the risks associated with soil biodiversity in your everyday practice. Some of these are unproven, others are well known in the scientific literature, but I thought I'd go through, oh, can you see that? Yeah, so I'd go through, I'm going to get down, go through just the ones that are highlighted, and I won't talk about pasture diversity because I think that's been covered. I won't talk about soil tests because that will take 20 minutes in its own right. So the first one is topsoil disturbance and exposure. I think I said the other day in the paddock, that's important because most of the diversity is in the top 10 centimetres. So we want to protect it as much as we can. And I believe that the rule of thumb is about 50% surface coverage will provide some protection. So if we can treat that as the bare minimum or the minimum standard to aim for, that's good. So these are just measurements that perhaps highlight the fact that that most of the populations, most of the diversity is in the top 10 centimetres. These are just increments, increments of 10, 20 and 30. And you can see after 10 centimetres, microbial biomass, the way that the populations just take a nosedive, there's nothing below 10 centimetres. And this will vary depending on your soil type. And the bottom one is a Shannon's index, which is an index of biodiversity, and that also declines. It's probably not as sensitive, but there is a decline in diversity as well, which we expect. So protect the soil, protect your population. The second risk that we're confronted with is one about compaction. We've heard also about that. It's just the squashing of the soil pores. So there's no pore space. And one thing I didn't mention is that the great biodiversity we support in our soils exists in different pores in the soil profile. So the smallest organisms, the bacteria, live in a really small pore. So if you've got a clay soil, they will inhabit the smallest pores there. And the larger organisms, which can be quite predatory on the smaller organisms, live in the larger pores. So there's a predator-prey thing going on as well, and that will happen if you've got open pores. But if you've got compaction, those pores have disappeared. This is just a graph over here of the bulk density readings and how that disappears when I point to it. Yeah. Is that because it's a screen? Oh dear. Yeah, so on the x-axis you've got the bulk density measures. I think it's up to 2. And then on the y-axis we've got the change of microbial biomass. So the blue line is 0. If the change is negative, it's below that, positive, it's above. This is a collation of 8 different reports or scientific papers, and generally when you get above 1.6 grams per cubic centimetre bulk density, most of the all of the changes are negative in the microbial biomass. There are quite a few negative measures or negative changes even at the 1.2 end, but by about 1.6 everything becomes they're negative changes. And I thought, well, how do you extrapolate that to field measures? Well, there is a relationship between penetrometer resistance and bulk density related to how much soil moisture you have. So you can use that as a calibration for measuring your bulk density and therefore the likely effects on microbial biomass. The next risk is waterlogging. We've heard all about it. The second farm we went to had some soils that were very heavy clays that were prone to waterlogging. And waterlogging just means that there's, you know, the pore spaces are filled with water. And water, or the availability of water, is a really important regulator of good microbial activity and bad microbial activity, if you like. So if you go through that green zone, from 10% water-filled pore space up to 60, water-filled pore space is the same as volumetric moisture that you measure in your sensors in the field, so it's roughly correlated. So the green zone is from 10 to 60% water-holding, water-filled pore space, and that is the zone where all the nice microbial processes occur. So the generation of nutrients, nitrification, carbon decomposition, mineralisation, all happens in that lower water-filled pore space level up until 60. So plant nutrient generation happens there. However, you reach a sweet spot or a point where things become more anaerobic after 60%, and therefore it switches on a whole range of other microbial processes that generate nutrients generate gases, so you get losses of nutrients from your system. So water and water-filled pore space has a real regulatory effect on what processes occur in the soil, whether you hold on to your oh my God. Okay, so established in the literature is the effect of urea on nitrogen fixation. It affects nodulation, so symbiotic nitrogen fixation, and free-living nitrogen fixation. There are reports of all different rates where you get an effect occur. In symbiotic relationships or symbiotic nitrogen fixation, the cut-off point is about 150 kgs of urea nitrogen, and then you don't see nodulation occurring. Now that's going to vary depending on where you are. It's even less in terms, the rate is smaller for free-living nitrogen fixers. They're more sensitive to urea. And it's because they're quite lazy. If there's a lot of nitrogen available for the plant, they don't need to do it. They can go and do something else. They can produce hormones. So they can switch their mechanisms, as I talked about before. There are, I'm going to skip the middle one. It just shows abundance of vesicular avascular mycorrhizae and the effect of phosphorus and nitrogen. And then there's new alternatives available, which I'll talk about in a little bit. I wanted to touch on seed coats because we don't know much about seed coats other than that they're coated commercially with a range of things: fungicides, insecticides, nematicides, etc., etc. And there's about 14 seed-coated products for grasses, brassicas and herbs in Australia. And I just posed the question after talking to Kate about what are the potential impacts and have they been measured? So is there a decline in rhizosphere biodiversity? Is there a disruption of plant signalling responses? A disruption of the recruitment mechanisms between the plant and the microbe? So there's quite a bit. Is there a disruption of nitrogen fixation, VAM colonisation? We don't know, but it's something that we should be looking at. Biologicals. One of the fastest growing areas in the production of agricultural products relates to the production of biological products. The biological product market is exploding at the moment. I think the figure is that now it's a $16 billion market, projected to be $34 billion in 10 years' time. So we're going to see a lot more biologicals on the market. If you split up, some work was done in 23 about the biological products in Australia. 45% were microbial, 15 were composite blends, then 13% are full mix, and there's a range of other things. If we split the pie another way, we can see that there's 33% soil biology stimulants. There's, I think, 20% black plant growth stimulants, etc., etc. So then if we look at how many of those products have been registered or put through a process of testing, very few have. And you can see here that 86% of those products are not registered, whereas only 14% of those products are registered. Now, I'm not promoting registration, but I think it affords some level of protection to consumers who want to know whether those products are reliable or not. Of the products that are registered, interestingly, they're all biopesticides. So, last slide. I started to ponder after speaking to Kate about wouldn't it be nice if we could do a few things in our systems. The first one is, wouldn't it be nice if the pasture could feed itself on the soils? We didn't have to add anything. The second one is, wouldn't it be nice if we could use artificial intelligence to help us predict what our pasture soils need? And that's not, it's not, we could definitely look at that as an option. It's not too far off. Wouldn't it be great if we could regulate some of our biological products through another body other than the APVMA? The second one is, the last one, I think we're already doing it. We're linking milk product quality to soil quality. There's been a few examples shown already, so I think we're already there. So I'll leave it there. Thank you.
I might jump down also as well. Hi, my name's Angela Avery. I'm works for Agriculture Victoria, and I've got the absolute honour to talk on behalf of our research team on a new project. You can see the team there. I'd also like to really thank the project committee, that being Jo Jacobs, who is the leader of the Dairy Feed-Based Program, Claire Hill, who's here today, who leads the Victorian Dairy Innovation Partnership, Kath Leskin and Donna Gibson, who are on our project committee as well. So thank you. You've been instrumental in helping form and shape this project. So Dairy Soil is a project, as I said, in the Feed-Based Initiative, which is a partnership between Dairy Australia, the Gardner Foundation and Agriculture Victoria. It is a fairly new project. We've been going for about 18 months, and we're really trying to link together what's been happening in the previous dairy feed base with new technology with a focus on soil, and it's just wonderful to be part of that project and to have soil in the feed base, which we've cheekily called Dairy Soil. Our study area is the Ellenbank Smart Farm, which you can see the details there. And it's good because we can start to then link the soil component to the mixed and resilient forages part that you heard Anna speak about yesterday, but also some of the dairy nutrition work as well. And it's also the same place where the Pasture Smarts was grounded as well, which we're leveraging off. So I guess Pauline finished with wouldn't it be good if. I'm going to start with wouldn't it be good if. Wouldn't it be good if we could actually measure our soil and manage our soil in the same way with the same resources and the same kind of technology that we do with pastures and put them together? And I reckon we're getting reasonably close to that, and we've got lots of opportunities to get even closer as we go forward. So I want to break down my presentation into some of the 4 areas that we're working in dairy soil. First, can we exploit paddock variability and soil profile variability? And we've heard a bit about how people are doing that practically already. Can we expand and extend the rhizosphere and resilient and mixed swards? We've heard about that. Can we use the next gen microbes as a microbial nutrient source? And Also, how do we retain and recover nutrients on farm? And I'm going to focus a little less on that today because of time. We've heard all the benefits that these things can deliver. Reduce fertiliser costs, reduce greenhouse gas emissions, reduce nutrient loss, improving nutrient cycling, improving water use efficiency, pasture production, and soil function and health. So good sense to do all that. So focusing on the first one, extending spatial variability, paddock to profile. And what we're sort of thinking here is let's get a really good understanding of the variability of the farm and the paddock. And I reckon we only need to do that once. So that's your underlying soil map. And we've got real good opportunities to do that with new technology. Let's take some very specific, nested, deep profile measurements. Just once, you might do that in another decade if you actually want to have a little bit of change. Then it's the regular soil sampling in line with key decision making. And Julian, I reckon we need to put a GPS on your spade and we'll be right, because then we can start to put it in a structural kind of thing across the farm. And then more real time around soil moisture, which we've heard. These can then be supported obviously for our variable rate fertiliser application, both soil and fertiliser and manure, the timing of nitrogen, a precise approach to multi-species, so let's match the species with the soil depth to topsoil, and let's start thinking about root architecture. We talked about what do we want to include in breeding programs. I'd be putting root architecture in there as well as a good one to really start to think about. And also, let's think about this pasture growth forecasting as well. So this is what we're doing in dairy soil. We're taking radar, spectral satellite data. We're validating it with gamma rays, I can never say that word, I'll let you do it, and EM38 data across the farm. We're doing deep core profiles at very targeted strategic locations based on that data. And we're also doing some work about how we can actually rapidly measure soil in situ. So we end up getting these kind of maps. So this is for the Ellenbank Smart Farm, where we start to get these layers. So we've got gamma ray draped over elevation. We've got EM38, we've got elevation, and then we've got our paddock plan. And the lines that are running through there are the soil profile that has been targeted in that system. So this forms your base underlay soil and map. And it'll align very closely to sort of some of the pasture growth maps and outputs you might get through Pasture Smarts. Also, you know, I'll just stick to this one, you get them not sort of structured as layers, you get individual maps, but they're also very important for other things. So your elevation, your topography of the farm is very useful to start to think about water across the farm, flow into dams, and you know in the southwest we're doing a lot of water dam infill modelling. So this can also support that. Once we get to that point, we can now start to ask the question: can we use satellites to get us real-time measurement in soil moisture? And we're not that far away from that. We've got the Sentinel satellites, we've got a new satellite coming up, and those from South Australia can be proud to note that South Australia launched the first satellite last year, and they're providing specific data in off-farm into that satellite. So there's going to be huge opportunities around what new satellites can tell us, and it's really timely and cost-effective. We get contiguous measurement across the farm, not just relying on where our soil moisture probes are, and we can get frequent measurements, and particularly at those shoulder times of the season where soils are wetting or drying. Really quickly, this is the type of approach we use, similar to the approach that validated Pasture Smarts. We use the satellite data, we combine it with the geophys, the elevation, the EM. We have soil moisture at all those points across the farm. We then measure the pasture samples, and we're also aligning that with Pasture Smart. So we're getting this really good map of soil moisture across space. I won't get into detail, but this is some of the imagery that you see we get from the satellite. We modify it down into a process, and we get these kind of quite detailed maps of soil moisture across a paddock. But we've also done it across a region as well. So this is southwest Victoria at one point in time, looking at variability. So this starts to give you a bit of an indication. We can say, well, why is a certain paddock got more soil moisture at this time and not the other? And in terms of this really helped livestock producers make decisions about whether they're going to put nitrogen on, whether they're going to sell stock early, all those sorts of things. So really good information. In dairy soil, we're also looking at rapid measurement through MIR of soil and really calibrating that against all the samples that we're pulling out from the Ellenbank farm, but also the history of catalogued soil we've got in Victoria. So we've got the handheld MIR, and you can see on the bottom there, that's the calibration that helps us calculate that out. That's for soil carbon, but we're also exploring with a new system, LIPS, which is used in the kind of mining industry, and they basically explode the particles, and then we can start to measure different components, their carbon, nutrient content. So imagine if you had something like this, maybe you not personally, but your agronomist could come in, and we've got our map, we've got our deep cores, and then we can come in and do some really quick, real-time measurement of soil to inform decision making. I should also say we're developing those same relationships up for measuring nutrient content in manure. The second area is expanding the rhizosphere and extending it, and I think we've covered a fair bit of that today. And yesterday. Our focus is very much the rhizosphere, because we believe that's kind of where it's all happening in this project, and people have described the rhizosphere, so I won't go into that. But we think it's very good and important for shoulder season production. So we've got some good modelling capacity, and we did this sort of pre-experimental model to ask the question that if we could increase perennial ryegrass root depth by 20% over a 10-year period, what might that mean in terms of soil moisture across the season? So you can, and the vertical axis is percentage increase in dry matter. So there's opportunity there to really start to improve dry matter. And the other thing I'd say about this is these sorts of tools, we know models never give you an answer, but it does give you a bit of a scope to support some decision making. So I was reflecting last night that, if you thought about Pam's decision making process, we could potentially start to feed in some of these scenarios to help with decision making around some of these key decisions. So in dairy soil, we're doing an experiment that is to start this autumn, where we're comparing perennial multi-species with a subsoil amelioration, and this is some work from Roger Armstrong in cropping, where you can actually see mineral N at depth. And the blue line there is where there's been a high proportion of subsoil, organic matter and manures applied to the subsoil. We're actually borrowing, or if people know Roger Armstrong, it's probably stealing, his bit of kit that's been used for a GRDC project. We're bringing that down to Ellenbank and using that as the mechanism to put in the treatments, which is pretty cool. And you can see there you start to get that slotted organic matter down at depth. One of the big areas that we're also working on in dairy soil is microbial nutrients, and Tim, Sawbridge and Tonga are in the audience today, so round them up for some specific questions. What we're doing here is we're actually sampling Anna's sites on the Ellenbank Smart Farm, and so the temperature and soil moisture data is what Anna presented yesterday. And you can see where we're sampling with the different coloured dots being the time of sampling before and after grazing and other management practices, so the paddock's been managed as per Anna's experiment. I think this is one of the first experiments that is really looking at the long-term and spatial kind of change in microbes over time, which we're pretty pleased to be able to do, and that will continue for another couple of years. So we are focusing on the rhizosphere and the roots. So we take a perennial plant out of the paddock and then look at both the root sample and the rhizosphere, which you can see at the bottom of that tube. And then we run analysis, in our case RNA analysis across that system, that plant. We've sampled 144 plants today and they've all been perennial ryegrass out of Anna's sites, but in addition we've done a couple from these other treatments as well. So what are we seeing? So this is the kind of first lot of roots. So on the first graph we've got the fungal type, and then the second we've got the bacteria. And you can see there's a lot of diversity there. There's also differences between what we find in the on the root and in the rhizosphere. And in addition, there's also differences between what we're seeing in paddock 1 and paddock 2. So there's a lot of diversity there. The brown there, as you can see, that there's, that's the fungi. And you can see there that, there's quite a difference between both the paddocks and the root and the rhizosphere. We've then got the nematodes mapped into those different treatments, as well as then over here the actobacteria. So really good way of getting an understanding of both what's there and potentially what is their function. So we think that, well, RNA will give us a really good understanding of what's happening, whereas DNA gives you a really good understanding of what's there, and Pauline was talking about sort of understanding what's there and what are they doing. So this graph here gives you an indication of a way of measuring the function. So this is one of the protein families that are linked to the ammonium and cycle. So this will help us really understand, what's delivering what and what we can do. So again, see the difference between the rhizosphere and the roots. We're also coming at this from a different aspect. So we're taking plants in the glass house and then to look at potential future microbes that could be introduced into these systems. And the good thing about it is by doing the research at the Ellenbank farm, we know what the background microbial population is, and that will give us an opportunity to introduce new microbe and bacteria. And so we've got some new ones that we're starting to see where we can build in higher dry matter, say with barley, and changing you apply it to the seed, and the phenotype is changed to improve things like dry matter production under drought conditions, and also with lucerne as well. So great opportunity there for really looking at evidence-based new microbial products. The final area I just really wanted to quickly talk about is what we're doing around the manure area, about retaining and recovering farm nutrients on farm. We're doing a few different experiments here. We're looking at how we can retain nutrients in manure. We're doing technologies around solid liquid separation of manure to really get a product that might be able to be applied more efficiently, but also through a subsoiling manuring type approach. And then looking at how it can be applied and measured both in the pile for nutrients, but also real time perhaps through the line as it's being applied. This is just some examples of where we've found what the value is of nutrients. And so up the top you've got 3 different regions, Gippsland, Southwest and North, and finding out what the different areas of nutrient content in manure. And the same with the bottom one, but we're also doing work on how we can actually map a manure stockpile and then think about what that structure looks like and the content in there. So, summary and key messages. Dairy soil is a really good way that we're kind of trying to bring it all together. So we've got spatial mapping of the paddock and the profile variability. We're combining that with remote sensing of soil moisture and hopefully soil type. And then we're looking at cheaper in situ measurements so we can supplement that. We're then really trying to understand the microbial rhizosphere and root diversity and function over time and over conditions, and then looking at lab identification of microbes and thinking about how that might be able to be reintroduced to solubilise phosphorus or add nitrogen. We're then considering how we can improve the rhizosphere to increase the depth and the extent of it through both subsoil amelioration and perennial multi-species pastures. And then finally looking at how can we actually get new kind of manure treatments and products to fall back into that. Putting it all together and building it in with a modelling function that is the same spatial modelling that underpins pasture smarts as well. So perhaps one day we can have a soil smarts and a pasture smarts, or one product that works together. So, take-home messages. It's really important, and we've got lots of opportunity to measure and manage soil variability to really start to unlock the benefits of precision technologies in dairy pastures, those that are available today and those that are going to be available in the future, and the future's not far away. Selecting the appropriate tools is really important, and we need to use a combination. Your eyes, the shovel. Let's try and put a framework and collect the data in a way that's spatial, temporal, and we can start to draw new knowledge from that. And soil by remote sensing is a real opportunity to start to predict pasture growth. Rhizosphere, biology of the rhizosphere is complex. We've heard that today before, and it's dependent on soil, species, weather, climate and inputs. Microbials have evolved very closely with the plant and with the root system, and thinking about them together is really important. And we really need to consider the remnant communities. Soil nutrients need to be efficient regardless of soil, and we need to improve our ability to manage them both in terms of on-farm and stopping nutrient loss. Just a quick advert. Australia's really kicked off its thinking about soil under the National Soil Strategy, and there's also the National Soil Action Plan. Tim and his team, we've got the opportunity to do a large soil biology project, so we're actually looking for sites to sample and do that same similar RNA testing. We're also doing it with Arthur Ryler, who are doing natural environments, so they hike up to the Grampians and Wilson's Pond to getting soil from there, and we're doing the same soil tests. So we'd be really keen to hear with anyone in the room who would be interested in us coming and doing that sort of sampling, and it would be fantastic if you had some remnant type vegetation that we could sample at the same time. Yes, there is a added thing that's gone on your table, which is the dairy soil little bit. It's got a QR code. If you want to follow the project team and follow the project, let's look into there. Thank you.
Good morning. Wow, what a heavy session to follow. So, take a massive breath and then let it out again. What we're going to try and talk about is really how do you fly without all that science that's been presented this morning to get from where you are right now to where you might want to be in terms of your soil health, your plant biodiversity, your soil biodiversity. How do you get thriving biodiverse life going on your farm? So yeah, I think we don't necessarily all have perfect knowledge, and we certainly don't have the resources that some of these research institutes have to fix the soil really quickly. Is the microphone cutting in and out for everyone? No, yeah, so Pete and I are just going to give you a little bit of a what have we done with the journey, and what have been our disasters, and what have been our things that we think were really useful or worked really well. And yeah, then just some of the summary of what we're seeing in the soil now. So we have already given you a bit of a summary of the things we stopped and some of the things we started. So I think in interest of time, I will just keep going on. So that some of the things we started there, we talked about them yesterday, but we're just going to give you now a bit of a timeline.
Alrighty, so before we started, 6 years ago, we were putting on about 80 kilos a hectare 6 times a year, so of urea, 220 kilos a hectare, putting on the pasture, a bit of muriate of potash, and the usual stuff, some DAP, seeds with fungicide, pesticides added. So in year one, when we first started on the journey, and one of the whys why we started on the journey was years ago, about 30 years ago even, I was working in a DPI in Tassie, and the benchmark there was 12 tonne of dry matter on an irrigated paddock, and there was no nitrogen involved. So we were putting on this nitrogen, we were getting our 12 tonne rate. It always sort of felt a bit like we weren't getting anything out of the nitrogen, and we had to go back to the old system. So that was always ticking away in my mind, and this is how we started the journey. We basically, after listening to Nicole Masters, adding a carbon source into your urea, she said you could basically drop your urea to 75% just by adding some folic acid of a kilo to the hectare. So we started doing that. It was a bit of a nuisance for our worker who was mixing it into that was as a solid at this stage. We got down to 165 in the first year with no change in plastic consumption.
So it's a really messy process. You just put your urea in your hopper as if you're going to spread it normally, and then you just sprinkle your folic acid powder. One of the things I think farmers find useful is we can just buy that at Elders in a 20-25 kilo bag, rip the bag open, sprinkle it over the urea. There's something very quick to put a carbon source with urea and start changing.
Yeah, so suddenly you're down to 75%. That's it. Go on, Pat. Yeah, next year we started skipping a bit of skipping rounds, trying to promote the clover a bit more, and we basically got rid of the MOP, which we thought was, or being told by everyone, with the chloride in there was killing our fungi, so we swapped over to potassium sulphate, even though it's more expensive, and we basically thought, oh, it's a lot dearer, but anyway, and then we were convinced by talking with Joel Williams and Nicole Masters that we meant to do that, so we did that. We're on 150 kilos that year. Yeah, a big drop then became when we started using the toll and vert machine and going to liquid applications of nitrogen.
Is everyone aware of a toll and vert machine? So it's basically a massive tank with a fantastic mixing device which helps solubilise almost, well, lots of things, not everything we thought about.
Yeah, you can use a boom spray, but it's a lot, with the advantage of the toll and vert is you can put a solid in there and it'll mix it in really well, so you don't need to buy things as a liquid, so it's a fair bit cheaper. And Joel Williams was saying to us that even by just going to putting a foliar on, you could drop your end use to 25% without getting any drop in production. So the same end is going to the plant if you use a foliar. So that's a bit of a good reason to do it, even though it's a lot more mucking around than bringing the phone and getting someone in to spread the urea. If you use a 25%, it's a fair incentive. In the time when we went to that, urea was sitting over $1,000 a tonne, so it was definitely worth our while. We never really actually put that high amount on though. We sort of were already on the way. The clothes were starting to really grow, and we can see that we didn't really need the high levels of urea, but we still just put a little bit on. And we're always just tickling it along with carbon source, sulphate of ammonia, that sort of thing, in the time for just small amounts, and it was more about stimulating biology than feeding the plant directly. And that was probably our big change, was moving to feeding biology rather than plants directly. So, yeah.
And the other part about using the foliar is we're trying to tell the plants, life's luxurious, photosynthesize like mad, grow and grow and grow, but we're telling the soil microorganisms, the rhizobia, the other organisms in the soil that will scavenge nutrients for the plant, that the conditions are a bit scarce. Please go about your work as best you can. And that if we were using granular, we would be doing the opposite. We'd be giving them a time when there's a luxurious amount of phosphorus and nitrogen particularly, which might turn them off and make them say, oh, well, we'll have a little holiday or we'll go and do something else.
Yeah, there was a question yesterday about putting on or reducing nitrogen and what was the advantage. And apart from the $100,000 bill that Simon had, for us it's about that when you put on a lot of urea onto the soil, you're creating a very high nitrogen environment which bacteria love. Fungi don't like that, so they tend to drop off and you end up with this bacteria dominant system, which ends up, in our case at least, with compacted soils. And you know, we've talked about that already today. People have talked about the problems with compacted soils, and not having fungi in the mix, they're not doing the job. They're doing a lot of jobs that for us, so basically our system is to promote more fungi.
So just on bare seeds, sorry, on that. On the bare seeds, that actually at the time was quite a challenge. If you just rang a seed company and said, can I buy blah blah seed, or you know, this particular mix or whatever we required, it automatically turned up coated with fungicides and herbicides. And I think it would be an exciting development in the industry if the default position was no seed coating and you requested the coating that you wanted. There may be cases where you might just want a fungicide, or you might just want a pesticide. And yeah, so finding bare seed at the time was actually challenging.
Right, I guess the thing we started using in year 4 was looking at doing our soil testing with the total availables in the soil as well as the readily available. So EAL up the back there, their labs will do that for you. And a lot of people will say, well, if I'm not putting on any fertiliser, how are my plants going to grow because they're not getting any nutrient? But where they're getting it from is the biology is sourcing it for you from the totals that are there. So we look at our totals, we look at our available, and when the totals are low as well as available, we'll start putting that sort of stuff on. We also need to put a little bit of the others on until the biology is getting to a stage where it can actively seek out the other stuff that we want, getting the phosphorus and the potassium from the totals, bringing it across into the available. So hence why we put a little bit of stuff on, and what we were generally low on, you can see that list of stuff there. Boron and the copper and lithium and Mg and everything was, well, Mg is very high for us, but the ones that are low, we put them on in the time for machine each time. This is a little bit to keep things ticking over, and then at the end of the year, in April, we've been putting on like a corrective, which is essentially a big boost of lime, gypsum, and all those things that's there as a solid. And one of our problems has been low calcium relative to magnesium, which also creates blocky soils. So we've been focusing a lot on getting the calcium levels up, which fungi also love high calcium soils. So that's what we've been doing there. And like that year, we got down to 29 kilograms of N to hectare, and with the same amount of plastic consumed.
So on that last slide, Peter mentioned the biological sequence. This is the sequence that we have been gifted, I guess, by Hugh Lovell, who is a fairly well-respected person who understood the interaction between minerals, microbes and plant growth. And so he talks a lot about if you don't have enough sulphur in your soil, sulphur acts at the edges of the soil colloids and makes the changes there. So that like the edges of those soil colloids is where we want the microbes to get going and start extracting particularly the phosphorus and other items off there. The boron and silicon are particularly important for moving the sugars up and down through the plant. So if the plant can photosynthesize but it doesn't have very good movement of the sugars, it can't send it down and out the roots to feed the microbes. So that's why they're so important to get going. And you can see extraordinary responses to boron. And all our soils across the McAllister are exceptionally deficient in boron and silicon. And that's been known for hundreds of years. So, yeah, so... beg your pardon.
4 minutes to go.
Thank you, sweetheart.
Right, so this is the year we're in last year. What else do we have there? Fish oil also. Well, that was something we put in that, and that's nothing that Joel Williams talks about. It's a protein source, so you're bypassing a lot of the processes that use energy by the plant, giving them straight into a protein, and it's again another. You can put a lot less on than you put a lot of urea to get the same result. So we only put on 2 kilograms, 5 litres a hectare of that when we put it out. And Kate actually made a bit of that a couple of times. Just when we pump out our water holes, the carp are there, and there's also carp, and Kate and one of our sons jump in there and they grab all the carp out with a net, chuck them in a wheelbarrow, and she chucks it in a big cauldron and makes up a brew of carp, molasses and milk. So if anyone's into that sort of thing, can you imagine what it smells like after a couple of days? Well, I can tell you it's not that great, but it does work. 2 minutes. So yeah, last year we were in the 20. That's sort of problem we had last year with a bit of flooding on the place.
Yeah, so we thought we were absolutely smashing it because we were continuing to increase our pasture consumption and drop our nitrogen and put a whole heap of different nutrients in, getting our plant diversity up. Things were going really well. And then we had this October flood, impacted a fair area of our farm. And, oh, that was a moving one. Yeah, so then year 6.
Oh, okay. This is your thing, eh?
Okay, so year 6, we knew we had an incident where we were going really well and suddenly we crashed milk production. We actually increased milk production, but we crashed pasture consumption. We didn't cut any silage and we had gone back to tighter, harder soils after that flooding. So it was a flooding event which would have been killing microbes because the soil was too wet for too long, causing denitrification, all the things that happen. Additionally, we've got to have to put the cows somewhere, so we caused compaction from putting the cows on the wet soils. So we were trying to think, well, what are we going to do to try and rapidly get ourselves back? One of the things that we learned about was a microbial mix called BAM, which is beneficial anaerobic microbes. They're actually facultative anaerobes, so they can live a little bit in anaerobic soil. The idea there was to introduce them into the system so that they could get down there and fix up whatever had happened due to the waterlogging. So yeah, we started brewing that and really brewing it just as a way to get from 20 litres to 1,000 litres in a very cost-effective manner, and started putting that out. And additionally with the BAM, I wanted to learn how to get our effluent. So the new pipeline came through our property also through that time, and that meant that our irrigation water for more than half our farm is now able to be delivered with pipeline. We're able to pump our effluent direct into the on-farm pipeline and take effluent over a much larger section of the farm. What we wanted to learn how to do is make that effluent an excellent source of nutrients for plants, rather than a source of nutrient that grows high nitrate feed and dock. And our understanding from that is that it needs digesting. It needs basically to be made into a liquid compost. And so we try and add in these beneficial anaerobic microbes. Again, anaerobic because an effluent pond is anaerobic. And
So we're out of time, unfortunately, we can't show you the whole of our presentation, but we can talk about later. But the a couple of things I just wanted to say to finish off, and one of the things that Matthew Evans said last night was talking about Dan Ketteridge's work on nutrient density of food, and that's something that we see as sort of a holy grail if we can show. And well, he's shown that the only thing that really gives you a high nutrient density is having a diversity of soil microbes, and it didn't matter what sort of farm system you had, if you had that right high density was there, and he's being able to show that there is a big range in density of food. If we can actually show that with our milk, suddenly we're going to get a fairly big uptake in the process. The other thing I wanted to say was, doing all this stuff, it's actually a lot of fun, and now as we talk about the fun factor, but you get up in the morning and you go out and have a look at what's going on, and it's awesome. So if you get into it, you'll probably enjoy it.
Ladies and gentlemen, what a jam-packed session. Gillian, Pauline, Angela, Kate Myrons and Peter Neves kicking things off with chocolate cake. How good was that? So I'll hand over to John Henry for our next part of the session, taking us through to lunch. Thanks, John.
Thanks very much, Greg, and good afternoon, everyone. Given what you have been able to listen to, reflect upon, ponder over the last day and a half, it was too good an opportunity to miss to use the collected people in this room to ask you to reflect upon and then write down your ideas for future collective effort in either research, development or extension. So the organising team, the Westvic team, are walking around right now and they're handing you out a piece of paper. The piece of paper looks like this. It's got a single question on it. Everyone in this room has a pen. More importantly, you've got a brain. So what we'd like you to do is we would like you to read the question and then write down your thoughts. Here is the question. What do Australian farmers need to know to increase confidence and application of soil health and function and multi-species pasture production? That's the question. One question. What we're going to do is we're going to ask you over the next 2 or 3 minutes, either by yourselves or through a little bit of conversation on your table, we would like you to write down what your thoughts are on that question, what your response is, and then for the last 15 minutes before we break for lunch, 20 minutes before lunch, if anyone is comfortable to put forward what they have written down, we're going to ask you to put your hand up, and then we're just going to go around the room. And we're going to try and collate we are going to collate all of these at the end. So that's the task. One question, you've got 4 minutes. Please write down your thoughts to that question, talk amongst yourselves, and then we'll come back as a group in 4 minutes' time. Ladies and gents, can I get you to, can I grab your attention, please? So just observing what was happening around the room, everyone was very diligently writing down their thoughts. So what we're going to do now, we've got basically 15 minutes, and we've got a bunch of people around the room armed with microphones ready to scurry their way to you. So I'm after some volunteers. Is there anyone who would like to put up their hand and who would be comfortable to share what they have written on their sheet? Anyone? Person right here, thank you very much.
How do we know that all this myriad of soil biological additives work, and what is the evidence that they do, and how can we prove this economically? But given that's probably going to be a really big task, is there a framework that we can evolve for farmers to actually make these assessments, or farmers' advisors to make these assessments in the paddock that they can actually use to evaluate these different products?
Okay, thank you very much, Tim. That's very well thought out. Thank you. Next one, up the back here.
Thanks, John. Probably lots of really good science, but really like to connect it from number 1 to number 10 or the circular, whatever it is, so that we have a really coherent pathway, particularly when we start talking about the wider community of dairy. And then secondly, connecting this bit into the carbon climate debate in a more formal way so that the science is included in, for instance, a carbon calculator, but there'll be other ones that I can't think of straight away.
Okay, thank you. Not sure who that young man is down the back, but thank you very much for that contribution. Oh, come on, that was worth a little laugh. One right here. Thanks, James. Got a microphone at the front. Oh, no, sorry, Tony, you're next. Okay.
I've got a topic, and then I've actually written down some answers for it. So I think you need to establish a pathway to start to move step by step to be successful. And then I've put down an option that people could consider. Step 1, increase the effluent pond size to capture as much water effluent as possible. See if there are any constraints in the paddocks that will be cultivated, example waterlogging or drainage, where that needs to be done first. 3, use the cultivation guide to help guarantee a successful planting. 4, use the effluent water strategically to reduce the risk of a dry spell or to start an autumn break, depending whether you're doing a summer or a winter planting. 5, model and improve grazing management strategy, extend the grazing time, maximise photosynthetic potential, maximise root development, maintain as much ground cover as possible, extend the growing season, and reduce stop fertiliser inputs.
Okay, thank you very much for that contribution, Tony. I think we were down the front here next. Thanks, Jade.
I think perhaps a centralised database of knowledge and evidence to create that pathway, as Tony said. So there's a lot of knowledge in the room, and there's a lot of knowledge elsewhere as well, but having a one-stop shop that farmers can go to easily sift through that knowledge and that evidence to create a really compelling case, I think would be beneficial. Highlighting especially the economic benefits or break-evens, I suppose, as well. It's either break-even or beneficial, so that really needs to be stressed. And perhaps a few more demonstrations and satellite sites so farmers can see local results as well.
Very comprehensive. Thank you, Jade. Down here on the left-hand side. Thank
Mine
you.
follows on from Jade's comment at the end, which is very much farmer-focused extension. So having peer-to-peer farmer groups run with on-farm demos, but use a regional champion farmer because they'll learn best from a neighbour when they're looking across the fence and what someone's done. So making sure it's on-farm and it's within a farming systems group or a grower group-led peer-to-peer group.
Thank you. Sorry, I can't see your name.
Brian.
Thank you very much. Yes, thank you very much for that.
Unless you're asking what her name was, and I don't think it's Brian.
What was asking?
Following on from Jade as well, the centralised data bank, thinking of how that can be utilised, we've got to be careful we don't end up getting a myriad of information and 10 hours later we realise we've got to go out and do our farm work. It's sort of, however, mindful of the structure, that it is something you can go to and hit your points and then explore them more as you need to. And so rather than each of us doing that, the centralised unit would allow us to, it gets done once, and it gets done for everybody, and we can follow a point, learn on that, and keep exploring further into that, and the network of links that go back on each other so that we can educate ourselves easier in our own time.
Thanks very much, Brian. Sorry, I had trouble. I was looking for you down here, but you're up the back. Thank you very much for that. We're off to now, Shaz. Okay, thank you.
Thank you. Farmers have basic knowledge of genetics, what multi-species are there, the environment and the management on their paddocks.
Thank you. Question on genetics. Thank you very much. Anyone else would like to enter the fray? Is it someone up the back first or? Okay. You can go.
What I want to add is just something about really valuing and encouraging the diversity of ways that people move forward rather than trying to just create one standard approach. Because what we've learned from ecology is the more diversity, the more likely that we get a population that can thrive in whatever comes next.
Thank you very much for that comment. Anyone else would like to? Richard?
We need to quantify where the nitrogen balance is. Where's it coming from? Are we extracting deep through the deeper roots? Are we fixing it? And how much has been lost in the environment compared to conventional, and how's that in synergy with our plants and our animals and the like. So I don't think we track the nitrogen.
Okay, thank you. Gentleman in the blue shirt, just here, halfway down.
I think the same thing applies to most of our nutrients that are going into that system. That in a high production system, a lot of it gets shifted around the farm, all that sort of stuff. So knowing what actual nutrients are going where, totals are sort of okay to a point, but it's about what's actually available. So monitoring that with both soil and a lot, especially for our trace elements, we've got to be looking at tissue testing because that's what plants actually are able to access. You know, soil testing for selenium and cobalt, good luck to be accurate with it. It needs to be tissued. And it needs to be a lab who can actually measure it too.
Thank you very much for that. I'm going to go, yes, sorry, let's go to Jack first, thank you.
I think the farmer message is that the science and the financials here are increasingly becoming more certain. You should have a closer look. Accept the diversity and pathways there.
Thank you, Jack. There was one down the back, Shiraz. Thank you.
I'll preface this by saying I'm not a farmer, but I guess synthesising the information that we've heard from the various presenters over the last few days, it looks to me like in the long term the benefits are clear, both from a financial perspective and then the resilience in a changing climate. But it also seems that it's not necessarily going to be an instant fix. So I think for farmers to know that it might get worse before it gets better is useful. And I guess Simon talked about the link to that. You need to have a financial plan to kind of ride that wave of bad, but then back up to good and something that's ultimately better. So knowing that in advance strikes me as something that would be useful for a farmer.
Thank you for that contribution, appreciate it. Just looking at the time, we've probably got another 5 minutes, folks. Are there any other things that...
Excuse me, I'm also not a farmer, so if I was going to do this, I would still be thinking about how I would be able to, say, select the right species. So finding out who is going to provide the right advice to me from that. So somebody has mentioned they had a great agronomist and they really valued that, but I don't know if you went just to your local seed company, would they give you the advice that was right for where you are, or, yeah. So I wouldn't know, do I need to do that, or do I just grab a bag of this and that and put it in and have a go? So that's a simple, non-farming person's point of view.
Thanks very much, Colin. Anyone else would like to offer their...
Yeah, I might have a crack.
Some Kiwi bloke down the front.
I think one of the fundamental things is, if we're going to give people confidence to do stuff around soil health, is what is soil health and how we're going to define it. Some simple measures that people could use. I think of the analogy of human health and the fine specimens such as yourself, John, on the stage there. We've got things that we can go to the doctor and get checked out. Richard tells me that his age, the doctor spends more time behind him than is in front of him. But, we got blood pressure, blood test, weight, circumference, all those sorts of things, and the steps that we take are individual to how we might become healthy people.
the same as every paddock's different, every farm's different. So it's not going to be one size fits all, but you've got similar metrics you can apply across that. And then you can look at cause and effect. You know, what if I stop eating as many potato chips like Lucy keeps telling me to? Am I going to get healthier and better? And that might work for me, might not work for Peter, or vice versa. So, you know, you can individualise it on a farm level, but having those simple metrics that everyone can aim for and can be on different parts of the journey. That's my thought.
Thanks. And I was wondering when proctology and soil health were going to intersect in this meeting, and it's just happened. So, well done. Thank you. Yes, sorry, Brad was there.
Do I have to declare that I am a farmer?
Thanks, Andrew. Yes.
I think from a farmer perspective, probably we need to develop a clear message on why is it working, why is multi-species working, and where's that nitrogen coming from, understanding that better. So, yeah, understanding the relationship between the different species that are in the sward and how they're interacting together, and, you know, for example, are they solubilising different nutrients and making them available to other species and working together like that, which leads to, Jay touched on the increase in the photosynthetic rate, which is driving the system. And I guess just understanding the full power of the rhizosphere, if we can get it working in a healthier state, and promoting that to the general farm base, I guess. And taking it, the challenge is always taking it from, you know, there's lots of us doing different things on farm, but taking it from anecdotal evidence and converting that to scientific evidence is one of the biggest challenges.
Thank you very much, Andrew. We've probably got time for two more. The young lady with the excellent hair colour.
Well, thank you very much for calling me young. I am a farmer, and I think my number one question, what I would want to know, is how is this going to affect my animals? Like, what does it do to my animals? Production, welfare, number one. The rest is great, but that's number one for me.
Thank you very much for that contribution. We've got time for one more, folks, and we're going to have to break for lunch. Gentleman down the back there with the beard.
I think what I contribute to the conversation is that in time we'll be forced to reduce high input farming, and we might as well try and find our own way forward, because we will be forced to reduce inputs.
Thank you very much.